Assalamu alaikum, dear student. Welcome back to the final class of the theory before the midterm exam. In this class, we will try to address few concepts about the deviation in the Mendel laws. Now, sometimes genotype does not always produce the expected results. For example, if we cross heterozygotes two individuals and we don't get the 3 to 1 ratio. So in that case, one phenomenon is although the Mendelian ratio is being followed, or the Mendelian laws are being followed, but there is incomplete penetrance, and that is basically percentage of the individuals having particular genotype genotype that express the expected phenotype. So it would be the percentage of the particular genotypes or individuals that have that particular genotypes and they are expressing the expected uh, phenotype, it will give us the incomplete penetrance. Example of this is the polydactyl. Generally, in human there are five fingers in toes, and in this case we have observed the six fingers instead of five. Expressivity, this is another concept. This is the degree to which character is expressed. Be the level of the expression of the genes, it is lesser or it's greater, that is going to determine to which that phenotype appears. And this could be the lesser or the greater effect could be due to the certain environmental factors or because of the interaction with the other genes in the system. So this is the photograph of the polydactyl, and you can count there are six fingers in each toe instead of five. Now, penetrance and expressivity. If we go for the karyotyping, then the Mendel principle of inheritance have lacked for example, the dominance. In certain cases, there is incomplete dominance. In certain cases, there are co-dominance. Then the multiple alleles, where certain sex-linked genes, they don't follow the classical calculation of the Mendel laws. And the polygenic inheritance, for example, the milk production in the cattle, that is basically a polygenic trait because it involves many genes. Then the epistasis means one gene is affecting the expression of the other genes that is located in another uh, locus. Then pleiotropy, one gene have multiple effects. Linkage, the genes are linked together, they always travel together and they don't follow the Mendel law. Then the transposons, these are the jumping elements which can change the expression of the genes. Then the mutations, certain mutations are there which can alter the frequency. And cytoplasmic inheritance, it does not follow the Mendelian laws. Now, lack of the dominance or the incomplete dominance, for example, you have familial hypercholesterolemia. This is the medical condition. In that case, there is incomplete dominance of dominant allele, in fact. Because of that, this problem appears. Similarly, the codominance, for example, in this case, the roan or pinkish brown phenotype is shown in the horses, and this is basically a mixture of red and white hairs, and that gives to the roan pattern of the skin color. This is the example of the codominance in the livestock. Then the multiple alleles, we know ABO system. There is no diallelic, rather it's multiple. There are three alleles involved. So that similarly, epistasis. I have just briefly about that. Interaction of two or more genes. For example, deafness in the human and the albinism in the human. So they are linked one. And this is called the epistasis. The one gene for the deafness it can affect the expression of the albinism gene and the vice versa. For example, in this case, if the genotype is like this, capital A, capital B, small b, or the vice versa, small a, small A, capital B, capital B. So both in this case are the deaf. But if they are intermarry, all their children can hear because they are heterozygotes. So in this case, basically one recessiveness or one dominance is going to affect the other gene's expression. And when they are in heterozygous condition, they are called normal. Pleiotropy, one gene can affect more than one trait. For example, sickle cell disease, it can glutamatic to valine. This is a change of the amino acid, and it can affect more than one trait. Then the polygenic inheritance, the group of genes after they get together to generate a phenotype. I have just given you example of the milk production or the height in a human being. So these are basically polygenic traits. Sex-linked genes. The X-linked genes are the sex-linked genes. Certain forms of the hemophilia. We studied example at the start of this chapter about the hemophilia. About the start of this course about hemophilia, and that was sex-linked. Then the red-green color blindness, then the baldness, then the Duchenne muscular dystrophy (DMD). These are basically sex-linked medical disorders. Then the chromosomal inheritance and the cytoplasmic inheritance. They have also their difference because chromosomes they are normally diallelic, but the cytoplasmics they have multiple copies, so they are you can say polyallelic. So their inheritance is not following the Mendel laws. And those which are located on the cytoplasm, in the present in the cytoplasm, in the mitochondria, in case of the animals or in case of the human being, so they express differently to the Mendel laws. And they are normally inherited from the mothers. The reason is that the father have very few copies number of the certain the mitochondria, while from the in the sperm, while in egg there are many copies. So generally their inheritance is from the mother. Reason being that the ovum have more mitochondrial copies. Some genes can modify the other, and in that case we have to go for the pedigrees, and we have studied already the pedigree. Here, for example, the dotted they indicate the affected one, and the blank they are normal. Uh, the common genetic disorders in the human they can be predicted even in the womb during the pregnancy, and we use the DNA fingerprinting technology for that. And through the help of the PCR machine, you can work out which copy is defective and which is the correct one. And for that, we take the Coriono-Willis sampling in the prenatal screening test, and that's all. So here are some assignments. I will give you these assignments according to their added numbers, and then also will give you the date on which you or before which you have to submit on the Google class. So that's all for the moment. And with the end of this, we have finished the midterm course for this animal breeding in genetics 202. Uh, and please go for the please go for this study of this course through this YouTube lecture or your Google Classroom and prepare well for the exam. Best of luck, and you can generate your queries on the Google Class if you have any, and we can address them um, from time to time. Stay home and stay safe. Thank you very much. Have a good time.